Hello everyone. Today we'll talk about the structure of the chloroplast. Now, first of all, you must know that we have already done it in the first chapter when we did the plant cells, that plastids, there are three types of plastids: chloroplast, chromoplast and leucoplast. I repeat, chloroplast, chromoplast and leucoplast. So since there are three types of chloroplast, today we are talking about chloroplast, a plastid which contains chlorophyll. A plastid which contains chlorophyll. Just remember that much. Now look over there. This is the structure of the chloroplast. One type of plastid which contains chlorophyll. So this is called plastid. Specifically, we call it a chloroplast. Now, leaves are the most suitable and well adapted organs to carry out photosynthesis. This is because of the following reasons. Now, why do photosynthesis take place in the leaves? Or rather, why are the leaves most suitable and well adapted for photosynthesis? Number one, you can see it on the screen. Leaves contain chlorophyll inside the chloroplast. Now, this is the structure of the chloroplast. Inside the chloroplast, we have a green coloring matter which is called chlorophyll. Now, chlorophyll is very necessary for photosynthesis. Without chlorophyll, photosynthesis cannot take place because chlorophyll absorbs solar energy. It absorbs light. Another reason why leaves are the most suitable and well adapted organs for photosynthesis is leaves contain minute pores called stomata through which carbon dioxide enters the leaves and oxygen is diffused out. So the exchange of gases in the leaves takes place through the stomata. So that is why there are two reasons. Once more we will talk about it. Why are the leaves most suitable for photosynthesis? Because number one, leaves contain chlorophyll and it is the only substance which can absorb sunlight and use it for manufacture of food. Next, leaves contain minute pores because you know that during photosynthesis we need carbon dioxide and whatever oxygen is produced during photosynthesis should be diffused out of the plant body so this takes place through the stomata in the leaves. So leaves contain minute pores called stomata through which carbon dioxide enters the leaves and oxygen is diffused out. Not only that, leaves are well arranged to receive maximum sunlight. Since it has a flat surface, a lot of solar energy falls on it which can be trapped. Is it okay? Now, since we are talking here about chloroplast, a plastid which contains chlorophyll, let us talk a little bit about it in detail. Chloroplasts in plants absorb light energy from the sun. So chloroplast, as you can see, the chloroplast which contains chlorophyll absorb the solar energy or the light energy from the sun. There may be thousands of chloroplasts in the leaves. Chloroplasts are double membrane organelles. So how many membranes do chloroplast have? It has double membrane. And the inner membrane of the chloroplast is folded into disc-shaped sacs called thylakoids. Okay? So we have disc-shaped structures which are known as thylakoids over here, as you can see. The thylakoids contain chloroplast and other pigments and are arranged in the stacks called grana or the grana. So these are the stacks which are known as grana, and this is actually the thylakoids. So thylakoids contain chlorophyll and other pigments and are arranged in stacks called grana. Grana in turn are connected to each other and lie in a gel-like substance called stroma. So you can see the stroma here. Is it okay? So thylakoids contain chlorophyll again and other pigments and are arranged in stacks called grana. So grana in turn are connected to each other and lie in a gel-like substance called stroma. So these are the grana and this is the stroma, and the entire thing is known as thylakoids. So this, we have to understand the structure of the chloroplast when we study photosynthesis, because this is the main organelle in a plant cell which absorbs sunlight, and this energy will be used in the manufacture of food by the plants. So next we will study is now some of the activities. Okay? So first we will talk about the starch test. We will talk about the starch test, the activity 1 that is in the book. Now, during photosynthesis, we all know that starch is produced. The food produced by the plants is the starch. Now, how do we know? How will we know the presence of starch in the leaves? So we do activity 1. We can look at the screen. What is the aim of the activity? To test the presence of starch in the leaves. Now, how do we go about it? You have to pluck a leaf from the plant which has been exposed to sunlight. That means for a few hours. That means photosynthesis has taken place in the plants, in the leaf, in the presence of sunlight. Put it in the test tube containing alcohol. You pluck the leaf, once more, you keep the potted plant in the sun for a few hours, let photosynthesis take place, and then you pluck the leaf from that particular plant which has been exposed to the sunlight for a few hours. Now, we put the leaf which we have just plucked out from the plant, we put it inside a test tube which contains alcohol. Now we put the test tube, we do not heat the test tube, the alcohol directly over the flame. So how do we heat the alcohol or the leaf and the leaf inside the test tube? How do we go about it? We put the test tube in the water bath. That means a beaker which contains hot water. Now the water will start boiling, and naturally the alcohol inside will also boiling the leaf. Now as the alcohol begins to boil, the chlorophyll disappears. That means the chlorophyll will dissolve in the alcohol and the leaf becomes colorless. Okay? The leaf becomes colorless. Once more, how do we go about it? We put the leaf inside the alcohol which is contained in the test tube. Now we put the test tube inside the water bath containing hot water and we boil it to decolorize the leaves. Now we take out the leaves and then we wash it off in cold water, and to make it soft also we wash it and the leaf also becomes soft. And then we put it in the petri dish over here. This is the petri dish. Now we put in the colorless leaves, we put a little bit of iodine, few drops of iodine, we put it in the leaves, in that decolorized leaf. Now, if there is starch present in the leaves, what will happen is it becomes blue black. Otherwise, it will become brown. So this is the starch test. This is the starch test. Once more. What is the activity here now? It is an activity to test whether there is starch in the leaf or not. What do we do? How do we go about it? We put the plant in the sun for a few hours, let the photosynthesis take place in the sunlight, and then after some time, after a few hours, we pluck the leaf from that particular plant and put it inside a test tube which contains alcohol. Now we have to boil the alcohol. We do not boil it directly over the flame because alcohol catches fire. So instead, we put it in the water which is contained in the beaker. And then when the water boils, the alcohol inside, because of the heat, the alcohol inside will also boil and decolorize the leaf. So why do we put the leaf in the alcohol? To decolorize the leaf. Now we take the leaf out after some time and then wash it off in cold water and then put it in a petri dish, and over the leaf, in the colorless, now the leaf becomes colorless, and over the colorless leaf, few drops of iodine is put. Now if there is a presence of chlorophyll, the starch, now what will happen? It will turn blue black in color. So that is the starch test. Okay. Now we go to another activity. Activity number 2. This is the activity to show that light is necessary for photosynthesis. Now what do we take? Again, the leaf in a plant. Okay. Now what do we do is we take our potted plant and keep it in the dark for the whole night, overnight, so that all the starch which is present in the leaf is used up. Now after let's say 12 hours in the dark, or maybe 24 hours in the dark, it depends upon the plant. Now we make sure that there is no starch in the leaves, because in the dark photosynthesis cannot take place and starch will not be present in the leaves. Now before the test, what do? Is we cover one of the leaf in the plant with black paper, both the sides. We cover the leaf, certain portion of the leaf with a black paper from both the sides, and keep it out, the whole plant, in the sun. Let photosynthesis take place. Now obviously photosynthesis will take place in all the leaves of that particular plant. Now that particular leaf, we pluck it off after a few hours. We pluck it off and do the starch test. Okay. Now what do we find in this test is the portions of the leaf which was not covered by the black paper. It turns blue black, as you can see, because it was exposed to light. The top and the lower portion of the leaf was exposed to light. It was not covered by the black paper. But now, when we do the starch test, this portion, the middle portion, will not turn blue black, because photosynthesis has not taken place in that portion of the leaf which was covered by black paper. Now there is water, there is chlorophyll, but why doesn't the photosynthesis take place in this particular portion? Because there is no light. So this experiment proves that light is necessary for photosynthesis. Okay. Now we come to the last activity. And this activity shows that chlorophyll is necessary for photosynthesis. What is necessary? Chlorophyll is necessary for photosynthesis. Now for this experiment, we take a plant which contains variegated leaf. You must have seen a particular leaf decorative plant. The leaves, the middle portion is yellow and the outer portion is green, or the middle portion is green and the outer portion is yellow. That means in that portion of the leaf which is yellow does not contain chlorophyll, whether it's inside or outwards. Okay. Now that is called a variegated leaf. Over here, the outer portion is yellow, green in color, which contains chlorophyll, and centrally it is pale in color, means it does not have any chlorophyll. Okay. We dig now again the same process. We put the plant in the dark for quite some time so that the photosynthesis cannot take place. Okay. We make sure that there is no starch at all in the leaves when it is kept in the dark. Next is, now we put that potted plant with a variegated leaf. We put it in the sunlight. Let the photosynthesis take place. Now, after a few hours, we do the starch test. We pluck one of the variegated leaf from the plant, and then we carry out the starch test. Now, what do we see in the starch test is the outer portion becomes blue black. During the starch test, the outer portions become outer portion becomes blue-black, and the central portion where there was no chlorophyll, which was pale in color, does not become blue-black. That means, since there is no starch in the central portion or the central core of the leaf, which was pale in color because there was no chlorophyll, and excuse me, this experiment proves that chlorophyll is necessary for photosynthesis. So the outer portion becomes blue-black because it had chlorophyll and the starch was produced here. The central portion does not turn blue-black. Why? Because the central portion was pale in color. It did not have any chlorophyll. So this experiment proves that light is necessary for photosynthesis. So all the activity 1, activity 2 and activity 3, please copy it down in your laboratory notebook, biology laboratory notebook neatly. You can use both the sides of the page, okay, without let us not waste any paper. So let us do it in both the sides. We can use both the sides of the page. Okay, thank you so much for today. Have a good day.